All right, so today we're going to take a little bit of an overview on cancer and some of its biology. And I'm going to go through a series of topics that are all interrelated, but they're nowhere near complete. So I want to preface this talk by saying that there's an awful lot of gaps and items that I'm leaving out having to do with cancer biology as a whole. First, going to talk about cancer and what it is, talk about some of the nomenclature, what's the difference between a tumor and a cancer, what's the difference between a benign and a malignant cancer, talk about chromosomal aberrations and mutations, tumor suppressors, proto-oncogenes, and oncogenes, signaling pathways, and a few other items. I'm hoping that we can get it at least a good ways past halfway in each one of these individual topics today, just to give you an idea of where I think we're going to end up time-wise. Just a couple of tidbits. This somehow got cut off, but 1 in 4 deaths are due to cancer. 1 in 7 deaths are due to lung cancer. I want to point out that lung cancer is the most common cancer in men, and breast cancer is the most common cancer in women. So we'll use that as sort of a foundation to start to build on. Okay, there it is. There are also 100 different forms of cancer. There's the rest of the stuff. Okay, I may not like this. I may end up bailing out of this. My formatting is a little bit weird. So I'm sorry for that.
Can you select all and go to animations and like remove animations? So like on the left, click a slide and do Control A. Does that work?
I don't think it was animations that did that. Okay, I did that.
I think it's also fine just to go through the slides like this. Like we can see the slide fine.
Okay, well that's even better for me. Okay, so I want to start with cancer and what it is. Cancer is a genetic disease, and it should always be thought of in those terms. And it's a genetic disease in which some of the cells of the body grow uncontrollably and spread to other parts of the body. It's caused by changes to the genes and also to the chromosomes that control cell function. And this is especially important and relative to how cells grow and how they divide. Most cancers are caused by a combination of factors and causes. They don't arise from a single unique event, and that only. Cancer arises from multiple hits to a given set of genome components. It arises from mutations in critical genes, and we'll be talking about what those critical genes are a bit later. It also results in changes in chromosome number and chromosome structure. These two things, mutations and changes in chromosome number, are hallmarks of cancer, and I'll be talking about those again a little bit more. There's also a number of things that contribute to cancer. They don't necessarily cause it, but they contribute to it. Those include spontaneous errors that occur as cells divide. This also includes mutation, spontaneous mutation. Damage to DNA that is caused by harmful substances in the environment. Tobacco smoke is one of those key substances that everybody knows about. So is ultraviolet radiation, X-rays, cosmic rays, and there's a bunch of other things that are out in the environment, and they contribute to cancer. There's also inherited genetic errors from our parents. That is a component that sometimes plays major roles in certain cancer conditions, and as that is going to be true in schwannomatosis as well. There's a number of things that can make major contributions to cancer, including certain viruses like the HPV virus, the HIV virus, and hepatitis A, B, and C in particular. And there's a lot of other things that contribute but do not necessarily cause cancer. These can include things like hormonal imbalance. The one that comes to mind at the moment, but there's a bunch of others. So again, they contribute to cancer. They put individuals in a situation where they are have an increased risk of developing cancer, presuming that the mutations and changes in chromosome numbers actually occur. A little bit of demographics, a little bit of statistics on incidence in the world. These are from the last statistics that I am aware of from worldwide statistics. In 2022, there was 20 million cancer cases that were newly diagnosed. And in that same year, almost 10 million people died of cancer. By 2050, the number of cases is predicted to increase substantially based solely on population growth. In 2004, in the US, there was about 125,000 deaths and almost 250,000 new cases just in that 1 year alone in the US. On average, the diagnosis age is around 70 years old. And if there's any good news in any of this, it is that the death rate has actually, from cancer, has actually been declining over time. So it's almost been cut in half from 1992 to 2022. Where everybody, I think, is hoping that, especially if you're in research, that trend continues to go down. And that affects nearly all of the various cancer types. So cancer itself, again, I'm going to go a few circles here, so please bear with me. Cancer is a multi-step process. It is genetic, and it is also evolutionary. And I'll talk about what I mean by that in a little bit further down. It involves multiple serial mutations and cycles of selection for cancer cells. This leads to a progressively increasing capacity for proliferation, invasion, and metastases. A very important point about cancers is they are not genetically uniform. There is a lot of variation at the genetic level in any given one cancer. The DNA content and the chromosome numbers are frequently not normal, nor are they consistent from cell to cell or region of a tumor to region of a tumor. Cancers, importantly, evolve solutions to selective pressures, including therapies. So if you try to cure them, they develop resistance. And that resistance gives them a selection advantage when you try to treat them again. And that is extremely problematic when it comes down to therapy, a point that we'll discuss at the end of the talk. There are multiple oncogenes and mutated tumor suppressor genes that act in concert to cause tumor progression. I'm going to talk about much more about oncogenes and tumor suppressors and what they are. But the point that I'm trying to drive at is that genes cooperate with each other. And it is via that cooperation frequently that the cells give rise to not only the tumor, but ultimately the metastases. It is the metastases that ends up killing people. I mentioned the fact that cancers have numerous genetic mutations in a tumor mass. The cells in the tumor mass are a collection of different genetic diseases. And this is a very important component of cancer biology. Cells in a given tumor are not uniform. And because they are not uniform, it causes a whole range of different problems when it comes down to therapy, because you're not treating a single unique disease, but you're treating a multiplicity of diseases, all encapsulated in one cancer. So what's the difference between cancer cells and normal cells? What do they do and do not do? What do cancer cells do? Well, they grow in the absence of signals telling them to grow. They grow independently of outside control. They ignore signals that normally tell cells to stop dividing or to die. They invade nearby tissues and spread to other areas of the body. That's metastases. The cancer cells can tell blood vessels to grow toward the tumors. They can control their own feeding behavior, in other words. They accumulate multiple changes in their chromosomes. They rely on different types of nutrients than normal cells. One of the key features, one of the earliest findings about cancer cell biology is that tumor cells use glucose differently than normal cells. It's a fundamental finding. Came about in the 1930s. Tumor cells or normal cells normally use mitochondrial oxidative phosphorylation to make ATP. Tumor cells, on the other hand, don't. They use a different process that is associated with glycolysis. It's a totally different way of using glucose and generating ATP requirements. One of the very important components of and differences between normal cells and cancer cells is that cancer cells develop processes to hide from the immune system. They also trick the immune system into helping them stay alive and grow. And this whole thing, I think, is what I'm trying to do is paint a picture of the fact that cancer cells, how to put this, are pretty tricky. And they develop strategies to stay alive in spite of many efforts to work against them. They overcome, survive, but it's to the detriment of the patient.
Let me interrupt there to talk a little bit more about metabolism. So there's a lot going on with metabolism. What I'll say is that some of the groundbreaking cancer research these days is about hijacking metabolism for cancers. What I'll say is that it's only recently been, relatively recently in the scientifically been appreciated how metabolism is so engaged. I think we've known for a while that, there is some kind of vascular recruitment, the recruitment of the capillaries and these other things. But that would be like how conventional cells are aerating and what metabolism they're using. It's not really appreciated what the alternates are. So, oxygen is supporting what type of metabolism, but what about metabolism in non-oxidative states? That's something very new that we're discovering. And I want you guys in your professional careers to pay attention to that, because even if it's a part of a co-therapy or it's a supplement, there's a lot of very interesting things you can do potentially. And I'm not swearing by any of these, but the point is that this is active research. but things like calorie restrictive diets, things that maybe would be simple to do while you're also doing other things in your chemotherapy, maybe it helps. I think there's some early evidence. It's not solid yet, and all cancers are different, but it's something I'm telling you to pay attention to is what is the role of metabolism and recruitment. I also wanted to give you a few more ideas here. So if it wasn't clear, you have this tumor mass, this big growing mass, the cells in the middle of that mass generally are going to need vasculature. They're going to need capillaries recruited into action to grow and provide nutrition to the cells inside the tumor. Now, there are cases, of course, where tumors are growing, there is poor vasculature, you get necrotic tissue. So, and that can cause all kinds of other issues. Sometimes that's the biggest problem of all, is that you have this big, massive dead tissue in the middle of the tumor. So that can happen. Of course, because of the selection pressures, the cancers are essentially incentivized or selected for doing good, aeration. So I want to give you guys an analogy that really helped me, which is if you look at the human embryo, the development from single cellular to there's early on, you just kind of divide, and then it starts to you go through a process where there is a directionality to your cells, and then with that directionality, you start to grow in some cells, but also some cells start to die away, giving you distinctions in the different cell layers. Basically, like imagine like when you don't start by growing fingers, you start by growing a whole hand that's connected, and then the cells in between your fingers die away, giving you some of that distinction. So you start to go through this kind of complex division process with all kinds of signaling cascades. Now your heartbeat starts after 22 days, roughly 22 days. That is incredibly early. Imagine from the point of conception to 22 days later, you have a heartbeat. That's extremely early. And I want to make clear that that's a physics necessity. Oxygen can only penetrate, we're talking about, the oxygen being released from the hemoglobin. I'm seeing here just ChatGPT, it can only go 1/1000th of a millimeter. I don't know the exact depth, but it's a very, very tiny amount, kind of this exponential die-off of how far oxygen can actually permeate into the tissue. So, you need this massive orchestration of capillary growth to be able to support any tissues. And so, you kind of reach a size limit. This is also where complex life versus single cellular life, you need tissues, you need organs and vasculature to become a large animal, and it is equivalent to become a tissue over a certain size. You need that recruitment. So again, pay attention to metabolism, pay attention to recruitment of the capillary. Some of the chemotherapies that are out there, their main mechanism is to make it harder for that vasculature recruitment to happen. They make it harder for the arteries, veins, capillaries to grow and penetrate into this new tissue. It's just one of the ingredients that we're playing with. So with that, I want to also take a pause. Are there any questions or comments so far? Olivia, is everything clear so far? Any questions?
Yeah, I think so.
Okay, sounds good. Paul, I'll let you continue then.
Okay. Matthew is correct that when you have a tumor mass, oxygen does not penetrate well into the middle of that given tumor. Similarly, glucose does not penetrate very well to the middle of the tumor. And similarly, yet again, cancer therapeutic drugs do not penetrate particularly well into the center of tumor masses either. And part of the reason for that is that tumors also build up another unique feature, which is a pressure gradient going from the center of the tumor mass to the outside. they secrete fluids, but the fluids are only going one way. They're always going out. Those tumors are not well vascularized, so they don't have good lymph flow. So there's lots of ramifications from just the overall structure of a tumor. What's the difference between a tumor and a cancer? Both of them, the tumors and the cancers, arise from single cells that begin to proliferate abnormally. Tumors may be either benign, that's to say they're not cancer, or cancerous. And by cancerous, I'm referring to a state of malignancy. Cancers, on the other hand, divide without control and invade tissues. They are malignant. Only malignant tumors are referred to as cancers. So from a medical point of view, that's the distinction between a tumor and a cancer. Cancers spread, they metastasize to other parts of the body. And they do that by either direct invasion, direct invasion, or invasion through the bloodstream and the lymph system. A couple of differences between benign and malignant tumors. Benign tumors grow very slowly. They have a well-defined capsule. I'll come back to that. They're not invasive. They are well differentiated. They look like adult tissues. They have a low mitotic index. By that I mean they have a low growth rate, and they do not metastasize. Malignant cancers, on the other hand, grow rapidly. They're not encapsulated. They are invasive. They are poorly differentiated. They have a high mitotic index. And most importantly, of course, they can spread distantly. That is to say, they can metastasize. A tumor capsule, to come back to that, is a defense mechanism that host tissues use to sort of wall off the tumor. They generate a capsule that is largely made out of fibrotic tissue, a lot of collagen, a lot of fibroblasts, and what the host cell is trying to do again is just wall off the tumor and keep it away from the rest of the body. That is a very common strategy on the part of the host to try to just restrict the growth of the tumor. Just some classification and nomenclature. Benign tumors are named according to the tissues from which they arise. And they have a prefix indicating what tissue they came from, and then an oma stuck at the end of it. So a lipoma from lipid or fat is a benign cancer that arises, or benign tumor that arises from fat cells. A fibroma comes from connective tissue, fibrous tissue. Neurofibroma, neuro from a nerve, grows obviously from nerves. Schwannoma comes from Schwann cells, and so on. Malignant tumors are always named according to the tissues where they arise. They are broadly categorized as either carcinomas, adenocarcinomas, sarcomas, lymphomas, or leukemias. Lymphomas and leukemias are associated, broadly speaking, with blood cells. Carcinomas and adenocarcinomas are associated with epithelial cells. The difference between the two is that our adenocarcinomas tend to retain their glandular-like morphology. It's a little bit more complicated than that, but that'll work for the time being. I'd like to move on to a new subject now, and that is cancer and their mutations in chromosomal abnormalities. Cancers, as I mentioned, arise from mutations of normal genes. And it is thought widely in the medical community that several mutations are needed to give rise to a cancer. The mutations that do occur frequently lead to larger changes which affect the chromosomes. And these mutations and chromosome abnormalities are both hallmarks of cancer and are often interlinked with each other. And both ultimately end up being directly involved in the uncontrolled cell growth that defines cancer as a disease. There are some common characteristics of chromosomes in cancer cells. And because it's a hallmark of cancer, I want to spend a little bit of time on it. These chromosomes in cancer cells display aneuploidy. That is the presence of an abnormal number of chromosomes. Remembering that human cells, your normal cells, have 46 chromosomes. Cancer cells, on the other hand, have a range of different chromosome numbers, and 40 to 60 is very common. This can exceed 100 chromosomes per cell or more in some cases, especially in very advanced cancers. So clearly, chromosome number is totally out of whack in cancer cells. This is a type of genetic instability. Again, this is another characteristic of cancer cells. They have a persistent loss, but also gain of entire chromosome and chromosome arm structures. They have large-scale rearrangements in their chromosome structure. And as I mentioned, they display heterogeneity. That is to say that the chromosome number is highly varied, and that variation can occur even within the same tumor or tumor mass. There is, of course, a price to pay for this, and that impacts clinical significance. All these changes in chromosome numbers, the aneuploidy, is associated with more aggressive tumors and worse clinical outcomes. There is also a direct impact on genes. So if you have extra or missing chromosomes, this can skew the activity of potentially hundreds or even thousands of different genes in a genome. So you can take the metabolism of a normal cell and make it even wackier by changing the chromosome number and thereby affecting the gene number. And from a tumor's perspective, this promotes survival and spread of the cancer cells.
Before you move on, let's take just a little reminder on just the basics of chromosomes and basic biology. I guess, let me pick on Olivia. Olivia, could you remind us how many copies of a chromosome do you have? Like chromosome 3, how many copies do you have? And how does that biology work from an inheritance perspective? Just briefly. And Olivia, you're muted.
Oh, sorry. Hold on. Let me finish typing something for a notes line.
Sure.
Okay, sorry. What was the question?
Yeah, so the question is actually two questions. One was, how many copies of a chromosome do you have? Like, for example, chromosome 3. Paul mentioned there being 46 total, but how many copies of a single type of chromosome do you have, and how does that work from an inheritance perspective?
Like in humans?
In humans.
There's 2 copies. Like one set from both from each parent.
That's right. That's right. And so just to remind people, when you have the zygotes and, the sperm and the egg, and they're carrying a half complement of what's going to be that total, 46. And so you might have 2 copies of a chromosome, and they could be different, right? And there's some, actually, it's more advanced than just you have your mom's or you have your dad's. There's actually a step involved where they line up the maternal and paternal DNA, and they'll actually swap things around a little bit. They'll do a little bit of a do-si-do, which is kind of a one rotates around the other, and they'll just kind of switch some things up. And so this, for example, gives rise to 2 siblings who come from the same genetic parents but have different features. The red and the blue and the green eyes were swapped. You kind of have a little bit of a switch around. So there's a little bit of a jumble that you do even within a single chromosome, but the bottom line is you have 2 chromosomes. And there are all kinds of disorders where, for example, you have a problem in one of the genes in one of the chromosomes, but the other one is still good, and maybe that's enough. Often that's enough, but not always, right? Because again, one of the ideas here is that the number of chromosomes you have, that's the number of copies of it you have, that's going to affect the rate of protein generation. So if you don't have enough of the right thing, or if you have the right thing being made at the same time as the wrong thing, we'll also just remember that there are gene promoters, gene inhibitors on the sequence, and all kinds of things are interacting with the promotion or inhibition of gene expression. So there is a lot going on. I just wanted to kind of run through that reminder for the purposes of this recording. But I guess, are there any questions so far?
I was gonna say that I don't know if Olivia and Aria, I don't know if Aria you're taking AP Bios, but we do go into a little bit of detail about like, especially how you were talking about how they cross over and like introduce genetic variation in a lot of ways. So we do go over a lot of the stuff that you said. So I think you probably have a good amount of background knowledge.
That's good. I mean, that's obviously assumed by Paul's presentation, but again, I don't know what you guys know, and it's for the fact of the matter is it doesn't even matter because we're going to record this for
Yeah,
future
yeah.
purposes. But no,
I
it's
mean, it's always good to get like a
Yeah,
recap
a
on
recap.
all of it.
and you'll probably see that Paul, we're only on slide 16 out of 70, that there is a ramp up of knowledge. And so we're going to build on these concepts. And I think one of the hardest parts, and I'm telling you this as an adult with years in cancer research and like at my current position, even I, to this period, have trouble remembering the fundamentals and factoring them in, because the biology is remembering, right? So if I forget about the recruitment of vasculature, I may forget that, but the response I'm getting in my drug screen study might not forget, right? And like that kind of thing where like it gets very chaotic. We're going to cover a lot of concepts, and I think as a human researcher, just feeling comfortable going back to the basics and just reminding ourselves, there's some advantage to that. It's okay to go back to fundamentals because there is a lot going on, and it all ends up being relevant. But Paul, I'll let you continue if there's no other question.
Okay, after that pause, then we're talking about chromosomes and some of the changes that occur in cancer. Chromosomal changes in cancer can affect very small parts of a chromosome or much larger ones. You can have structural changes and rearrangements within the cancers that affect deletions, and you can refer to the cartoon that's on the left there. You can have duplications of genetic material, inversions, and translocations. Now, out of all of these different things that can occur, duplications are particularly bad because they can give rise to an enhanced number of proteins being secreted. And we'll get in a while, we'll get to why that is particularly bad. But just for the moment, remember that anytime you have a duplication of genetic material, that can have very bad consequences. These types of changes are very common, and they're found, for example, in over 90% of solid tumors. And they are frequently occur again within the same cancer cell. Not all the time, but they are frequently occur together. We're going to switch gears and go on to mutations. Mutations are induced changes in the DNA sequence. These types of changes can alter DNA function. They can alter the function of genes. That alteration in the function of genes, in turn, can affect and change protein structure and its function. And that, again, in turn, can then affect cell growth, division, and survival. So you end up starting with a very small change in DNA sequence that can even affect just one nucleotide sequence, for example, and you can have a very rapidly cascading effect, ending up on or impacting cell growth and survival. But I want to make sure that we put this into perspective. Mutations are a normal part of how cells live, and most mutations that occur in our bodies are actually repaired. There's an average of about 175 mutations per cell generation. So every time a cell divides, you get about 175 mutations normally. Again, most of these are repaired, and most of them don't do any damage. They're sort of a baseline of activity in a normal human cell. What that means is potentially you could have millions of mutations across an individual human lifetime. Again, most of these are repaired. The average person has around 400 defects in his or her gene and genome just walking around today. The average person inherits about 100 to 200 mutations from their parents. So I'm blaming it on mom. I'm sorry, couldn't resist. Most of these mutations are harmless and have no apparent effect on our health and overall appearance. But there is an underlying mutational profile that goes on with normal cell division and cells being alive. Again, most of them are not detrimental. There are a number of different mutations that can occur. I'll just go through these very quickly. There's point mutations, which affects a single nucleotide in a DNA sequence. There's insertions or deletions. There's translocations, things switching places. And there's amplifications, which are duplications of DNA segments. And of particular import is that you need several mutations serially to give rise to a cancer. You have accumulated damage over a period of time. These serial mutations lead to an increased selection within the evolving tumor mass, and the tumor cells are being selected for the most survivable and the most cancerous cells. Those are the ones that continue to survive and cause harm. And they are a result, again, of multiple serial mutation sequences. Where do these mutations come from? They arise from a combination of factors. There's errors that normally we talked about, normally occur during DNA replication and cell division. There's environmental factors, smoking, UV radiation, viruses, inherited genetic traits from a parent. All of these are predispose individuals to cancer and increase the rate and risk of subsequent cancer diseases. Since we're going to be talking about schwannomatosis, I want to introduce briefly, a discussion on alleles and mutations. So we had earlier you were talking about the normal chromosomes and how there's two pairs. On the left-hand side here is an illustration of a chromosome in two pairs. And for each segment here in yellow is trying to illustrate an allele. An allele is an alternative form of a gene that determines a specific trait. And typically there's one of these alleles on each individual chromosome pair. If you then have a point mutation, and here what we're doing is we're changing this T to a G nucleotide, to guanine, and that changes the amino acid sequence that comes out of it from a cysteine to an arginine amino acid. And while you're changing only one amino acid here, this change from cysteine to arginine is on a molecular level, is a very, very dramatic difference. It's taking something that most of the time does not have a charge, which is the cysteine, and turning it into an arginine, which is a very basic amino acid in its side chain. That can have profound alterations in the overall three-dimensional structure of the protein. Importantly, because there are two alleles, you can have damage to one or both. You can have a monoallelic mutation, which just affects one allele, or you can have a biallelic mutation, which affects the same gene. They don't necessarily have to occur in exactly the same place or exactly the same way. They can still affect the same gene and have slightly different targets at the DNA level. But not all mutations are created equal.
Well, can I interrupt? Is that okay?
Yeah, of course.
Okay.
Don't ask, just jump in.
Okay. Could you go back to the last slide? I want to do just a knowledge check. Aria, are you familiar with amino acids? Have you encountered that before?
Yeah, I'm taking AP Bio right now. We were just doing the biochemistry unit.
Okay, great. Can you tell us a little bit about kind of the sequence of how a protein is made? Like, what's your understanding for that?
So it starts with the primary level where there's a sequence of amino acids joined together by peptide bonds. And then there's the secondary structure where alpha helices and beta sheets are formed through like hydrogen bonds. And then there's the tertiary structure where like the polypeptide chain folds through like interactions like the disulfide bond, hydrophobic interactions, van der Waals forces, ionic bonds. And then finally there's the quaternary structure where multiple polypeptide chains like fold together to make like a functional protein.
Thank you. That's a lot on the structural side. That's very helpful. I'll also add that there is a glucoprotein processing that I would say is probably still not fully understood. Glucoproteins, they're adding these salt groups. Sugar groups. Glucose, sucrose. I don't remember all the details, but it's one of the ways that when your protein is being made by your cell, it's kind of being labeled and flagged as a byproduct of your body. Go ahead, Anushka.
I don't know if you're talking about the same thing, but in AP Bio, we studied a little bit about glycoproteins. Is that what you're referring to?
I believe it is. Yes.
Yes,
Because
it's really,
it like labels, like they act as labels, and I think, but we didn't go into a lot of detail. But just for everybody else, that's you might have talked about that in class.
Paul, did you have a comment there?
just that you were in fact referring to glycoproteins.
Now, what I'll
Now,
say
you
is,
also find the sugars in more elaborate structures that are outside the cell called glycosaminoglycans, but that's a story for another day.
what I'll say is that I believe, it's my opinion, that the science around what those are doing is lacking. Like labeling it, I think that's underselling its impact. I believe there's more. I believe there's more as it relates to cancer therapies and normal cell function. So maybe another thing to look out for in line with metabolism. Just think about it, be aware of it. The point that I did want to make is, however, the flow. Thank you so much, Ari, for going through the structure. And you're right, like the water, like the amino acids compress in the aqueous solution inside the cell. It's kind of a salty solution inside the cell. And the polar and non-polar regions kind of interact and form structures, and those higher level structures we'll call the what you were calling the quaternary structure, which was protein to protein. I call that a complex. It's a protein complex where things come together. For example, enzymes might bond with other proteins and maybe turn the protein on or off, and that would, when it's bound, it would be considered a complex. The flow I was getting at with the question was also talking about, you start with things going into the nucleus. So you're trying to turn on some protein production. So something is going into the nucleus. These are signaling factors going in. A lot of the signaling factors, what they're doing is they're finding and binding regions on the DNA sequence. A lot of what they're doing then is unwinding the DNA. So the DNA is tightly wound around these histones, and it's this very jumbled. It used to be like similar to like phone cords from back in the day, but just imagine like a spool of yarn all knotted up. It's very tight. It's very hard, and you can't really get the replication proteins in there. They just don't fit. So a lot of what these promoters are doing is they're binding to regions of DNA, unwinding that aspect, and just making it easier for the replication machinery to get to that part of the DNA and bind it. So a lot of the regulation, we talk about cell growth, cell promotion, is making it easier for the proteins that are doing the expression to be unwound, or conversely interrupting things that make that, so there's and then there's, of course, many dimensions in genetics. There's regions that promote. You can also bind a region and be an inhibitor. So if you bind something that would normally be opening up, but then you don't let go, you're actually inhibiting the production of DNA, or sorry, the introduction of the mRNA. I'm not going to get into the tRNA, mRNA stuff. There's tRNA becomes mRNA, becomes the protein amino acid sequence, and the amino acids go that sequence, the mRNA does leave the nucleus. It goes through the rough endoplasmic reticulum. I think, remind me, I think the smooth endoplasmic reticulum is involved for the glycoproteins, or was that in the rough? Does anyone remember?
It's in the smooth.
It's in the smooth. Okay. And I think the smooth endoplasmic it was one of the last steps. Now, the proteins have a lot of things that they're doing. A lot of times they are transported to a part of the cell. They're often in these like little vesicles, little cell lipid bubbles, and they're actually like walked to various portions, maybe the cell surface. However, they might be also just interstitial. They might be in the middle of the fluid. Some are in the middle. A lot are in the cell wall. A lot of things, especially the hydrophobic ones or the ion channels, like they're in the cell wall. The receptors are all in the cell wall. So they just kind of go all over. But I did want to give that basic, again, just preface, because that's a machine, right? You're talking about a machine that has orchestrations. A lot of cancer therapies are all about preventing division. And when you look at this complex machine, both from a promoter's perspective, an inhibitor's perspective, or just from breaking down cell division, there are a lot of opportunities to break it down and a lot of ways to do it. And a lot of those, in fact, are done, right? There's a huge variation in therapeutic styles, some having to do with the nucleus, some having to do with after it starts dividing and you have the mitotic spindle, just making it so it can't split, making that mitotic spindle not be able to break apart. Things like that. There's a lot of different styles in this orchestration. And like, I think it's part of the beauty of actually studying cancer biology. You kind of take it for granted how the cell is so elegantly wound. But then when you're studying the cancer, of how various aspects of that are either breaking or taking advantage, so a certain kind of appreciation for every step along the way, including, that there is this network that proteins can walk on, these spindles or something. I can't remember, but they're these protein path, highways inside the cell. Very interesting. Aria, are those concepts things you've encountered before, or some of that kind of new?
I've heard of some of the stuff you're talking about.
Okay, excellent. Are there any other questions or comments? Okay, Paul, thank you so much.
All right, I'm going to come back and say that not all mutations are created equal, especially when it comes to do with cancers. I want to talk about driver mutations in particular, because driver mutations confer a growth advantage. Collectively, these driver mutations, when you're talking about a cancer, don't occur in a huge abundance. They typically range between 2 and 10 driver mutations per specific tumor cell. That can occur in a larger array of mutations in the cancer cell itself, so that the mutations in a cancer cell as a whole can be much larger than the ones in driver mutations. So a typical cancer cell is going to have between 10 and 40 mutations, but the total number of mutations per individual cancer cell can range quite high, up to and beyond 1,600 mutations per cell. And yet 2 to 10 of those are the driver mutations, the ones that actually cause the cancer. The others, the ones that are not driver mutations, are classified as being quote unquote passenger mutations. The mutations are present, but they do not necessarily directly contribute to cell and cancer growth. Oh, incidentally, maybe you have been seeing these little things that I'm not talking about. So these are specific references that I included in a lot of the slides. These are indications of where I got the information that I'm talking about, so that if you want to look more into the subject, for any reason, you have a place to go that is going to be specifically where I got this information on. And you, again, just in case you have an interest. But it's also a way of keeping me honest, so that I'm providing you information that is coming from the scientific literature, scientific and medical literature.
And Olivia, as a point of note, maybe we could extract those references into your formal notes and include them as some of the reference materials to supplement this video.
Sure, that'd be good. Probably easiest is just whenever we get the slides. I
Yeah,
can
we'll send you the slides. I'll send. I think the PDF I can, the PowerPoint I can share that. So I'll make that available to you.
Awesome. Okay, thank you.
Thanks.
So we talked about cancer drivers. Well, what's actually being mutated here? And what is being specifically mutated that are being cancer drivers are oncogenes and tumor suppressors, which I told you we'd get back to. So this is just a graphic that that illustrates the frequency of tumor suppressors across a population of lung cancer adenocarcinomas. And what I've done is in red, point out the tumor suppressors, and in yellow, the oncogenes. And you can see that as an incidence level, both the oncogenes and tumor suppressors are quite well represented. These, in fact, represent the highest percentage of driver mutations in a given cancer. They have a lot of funky little names over here, that some of which we'll be talking about. TP53 is a gene that is involved in cell growth and its control. And it is far and away the most mutated gene in cancers. And it is a tumor suppressor. That is followed here by a different gene called KRAS. KRAS is, again, the most frequently mutated oncogene in cancers. So it's not surprising that in lung cancers, these are the two top dogs. So when we talk about cancer drivers, what are we really talking about? We're talking about oncogenes, tumor suppressors, and DNA repair genes. So to start with, I want to start with a proto-oncogene. A proto-oncogene is a gene that is found in normal cells. When it is mutated, it gives rise to an oncogene. You have to have that transition. Oncogenes are involved in promoting cell proliferation, inhibiting programmed cell death, which is also known as apoptosis, and increasing angiogenesis, which is the formation of new blood vessels. Tumor suppressor genes, on the other hand, are typically associated with a loss of function. Their function is knocked. You have a protein that, when it's a tumor suppressor gene is mutated, that protein loses its ability to function. It has a loss of function. And that is important because it releases control on things like cell proliferation. And it also, on the other hand, blocks apoptosis. So you have the worst of two worlds going on. And then also tumor repair genes. If you normally have a background level of mutation and you can't repair them, things are not going to be good. So collectively, oncogenes, tumor suppressor genes, and DNA repair genes are collectively referred to as cancer drivers. And yeah, I'll just leave it at that.
Let's take a pause. If you could go back to that last slide. Anushka, are you with us? you able to talk?
Yeah, I'm here.
Okay. Do you know the difference between proto-oncogene and oncogene?
I think so.
You want to give it a shot, or if not, I can certainly go through it.
I can try and see if I'm right. So I think as Paul was talking, proto-oncogenes is I know that they get activated to become oncogenes in like cancer cells. That's pretty much how much I know.
That's right. So they're the classic. They're the same gene. It's the gene that is an oncogene is also the proto-oncogene. But the proto-oncogene is that gene in its non-driving state. So in its normal state, its wild type state, and then once it becomes mutated so that it is being a driver in the tumor, then it's defined as being an oncogene. So the proto, I believe, just means, well, in this case, before, or archetype. Paul, do you have any flavor you want to add to that?
No, it's both of you are correct.
Okay, thank you. Any other questions? Go ahead.
Yeah, I had a quick question. So, proto-oncogenes can like become mutated to give rise to oncogenes. Can it go the opposite way? Like, is it possible for oncogenes to be mutated to like stop driving like cancer production?
Theoretically, yes. On a practical level, no.
Okay. Okay.
Usually what happens for most of the oncogenes is the type of mutation that is induced causes an amplification within the DNA sequence. So instead of having just one mutation that is giving rise to turning that protein on, you have more than one signal that is turning that protein on. So it's very hard to then go backwards. You're running against the stream of water, as it were, trying to get back the other way.
I see. I think I might have confused myself with I remember talking about like P53 in one of my classes, but I think that's a tumor suppressor gene and not an oncogene.
TP53 is in fact a tumor suppressor gene. And probably what you're thinking about is the fact that there are certain types of therapies that are trying to force the regulation of the level of TP53 to go up instead of down. But that's a unique approach that is specifically focused around tumor suppressors. And not all tumor suppressors, just a very few handful, and only one that looks like it has potential therapeutic implications, and that is TP53. But the oncogenes in general, they don't work like that.
Okay. Okay. Thank you for clearing that doubt.
Okay, Paul, I'll let you continue. I know you're in the middle of defining.
Actually,
go ahead.
I'd really like you to, Matthew, to talk about this one because you do a much better job of it than I do. Could you give this a whirl?
I'll try it. The analogy here is driving a car, right? And so we're looking at, let's say, the function on the left. Let's just focus only on the picture on the left. These are normal genes, non-tumor tissue, normal healthy tissue. The function that the tumor suppressors are doing is like the brake. So this would be an interruption of a process that's going on. So imagine, for example, you have a wound, right? Now, a wound is a normal biological function. It's going to create growth factors. These are things that are being secreted by the white blood cells and you have these epithelial cells maybe forming the scab tissue, and you have this recovery process. Now, that recovery process is supposed to encourage cell division because you're repairing tissue. Now, at some point, there needs to be an understanding within the cells, at least from a physics perspective, that the signaling for growth is terminating. So at all points for all cells that are going through this promoted cell division process, there has to be an off cascade, something that says no more. And so those would be kind of a lot of the functions of the tumor suppressors, that braking action of preventing the cascade of signals that is doing the cell division. But on the opposite side, if you talk about what is the role of the proto-oncogenes, it's the accelerator. So those are the things that, I'm receiving this growth factor and I am turning on the RAS cascade. This is a cell signaling, enzyme signaling cascade, resulting in things going into the nucleus, turning on and promoting protein production related to cell division. And again, that's the accelerator. Now, if we look at the mutated case, now going to the two on the right, if you have a mutation in the tumor suppressor, it's like the broken break, right? So something, gave you that growth factor. Maybe you got a scar or a wound, it's normal, it happens. So you have this growth factor, it's at the outside of the cell, it's noticed by a receptor in the cell membrane, and it's sending this on signal and turning on some kind of cell growth recovery, cell division signaling process. Now, with the tumor suppressor broken, that would be maybe considered a loss of function. It's not working, it can no longer do what it's going to do, so your brake is broken. So, you have the something being signaled and you have no way of turning it off, or it's just an impaired ability to turn it off. Either of those cases can result in rapid cell division, and that tumor can now become a very commanding survival force. Now that starts, that's the early formation of the tumor can get out of hand, where now with rapid division, you're perhaps introducing more and more mutations and more mutation pressure. That could also result in an accumulation of mutations, leading to the more malignant form. So just having just the broken break, just the broken tumor suppressor, that might be enough fodder to grow a tumor and for that tumor to naturally be selected and evolve for worse and worse accumulations of mutation. The oncogene here, the analogy is that you do have the gas floored all the way to the ground. So this would be, for example, that some part of that on signal, say it was turned on normally, maybe there's something about it where, for example, it binds to some kind of activating complex. So you have two things coming together they're in this activated state. Now maybe they're supposed to unbind. The normal thing they would do is unbind, but because of a mutation, now they come together to bind and they never undock. They're really sticky and they stick together in this on configuration. That would be like having the accelerator on your car stuck all the way down, running and running and running. So they both can result in cell growth and proliferation. And again, I really like this brake versus accelerator analogy. The tumor suppressors, which by the way are a lot of the research the adult working group is focusing on. We're specializing right now in tumor suppressors because a lot of times they are loss of function when they're mutated on both alleles. So that's to say that they're totally broken and you know, you have the pedal is down and there's no brake to turn off the cell division and propagation. So let me challenge Olivia, based on what you've heard Paul and I say, can you give your own definition of tumor suppressor oncogene? Do you have a way of communicating that?
Maybe.
We'll give you a minute on the audio issue, and if it's an issue, we can move on. We'll give her a second.
Is this any better?
No, normally with Echo on your two devices, one needs to be muted and volume down. Like you need to do both, volume down and muted on your secondary device. I'm assuming you joined in two tabs on purpose. If not, I can kick one of your tabs.
Does this work?
Yeah.
Okay.
Yep.
Sorry. Can you repeat the question again then?
Yeah, absolutely. So I was wondering if, in your words, do you feel comfortable telling the difference between a tumor suppressor and an oncogene?
Maybe. So one is, it's like with the car analogy, we have the like brake and accelerator. So the tumor suppressor gene is more of like a stop on the cell growth. And so when it's working, cells don't overproduce basically. But when they are defective, like typically they might be responsible for like apoptosis or that sort of thing. But when they're defective, the cells can continue to proliferate. And then oncogenes more are of like the cell proliferation itself. Like they cause cell growth to happen. And so when they are, again, defective, it's like overgrowth, if that's right.
Yeah, that's roughly right. Again, I like this brake accelerator analogy, so anything within that realm is at least equal to my understanding. I think Paul actually has a better way of saying it, but
No, it works perfectly well, and the concept is very clear. I've always liked it. It's a very clear concept.
okay. Any other questions or comments? All right, Paul, we'll let you have it.
Okay, so one of the perhaps natural questions to ask is, do these tumor suppressors, since they're so important, are they found in a lot of cancers? And the short answer is, yeah, they are. So if you look at just TP53, for example, as I mentioned, it's involved in DNA damage control, regulating cell cycle control and apoptosis. But it is found in most cancers, including lung, breast, colon, brain, and pancreatic cancers. RB1, which regulates cell cycle, is found also in childhood diseases as well, childhood cancers, as well as lung and prostate cancers. In women, this is a tumor suppressor of major concern. It is associated with DNA repair and affects incidence of breast and ovarian cancers. There are others that, for example, APC, which affects colorectal cancer. This is a signaling, this is involved in cell signaling, and controls growth and survival, and you find it again in lots of different cancer types. And the same is true all the way down. NF2, which we've talked a little about a little bit previously, is associated with cell growth. So is NF1. And you find NF2 in schwannomas and meningiomas, which are tumors of the nervous system. We'll be coming back to these in another project later on. And there's others. So these are a list of some of the common oncogenes that you see and their associated cancers. It's sort of the same story. You have RAS. These are all named largely as abbreviations. They do have full names. Some of them are very short and some of them are very long and complicated. And some of them may not mean a whole lot to you. RAS originally came from rat sarcoma, which is where this gene was originally detected. And these are two, the KRAS and NRAS are just variations that came out of that. EGFR, this is a gene that makes a protein called epidermal growth factor receptor. And they all have full names, but they tend to be just abbreviated in shortened codes because the full names are extremely large and very difficult to remember for anybody. The point that I'm driving at, however, is that these are some of the major actors in terms of oncogenes, and they affect a large number of different cancers. And importantly, frequently, the tumor suppressors and oncogenes overlap in an individual tumor cancer. So you can take a bad situation and make it worse by having both of them in the same tumor, especially if they're in the same signaling pathway. Signaling pathways are something that we'll talk about next, or a little bit later on in more detail. Again, we come back to cancers arise as abnormalities in genomic control and they affect cell processes. Both the oncogenes and tumor suppressors affect molecular pathways. Specifically, signaling pathways that control cell proliferation, apoptosis, and DNA repair pathways. Again, sort of a common theme. They're always affecting the same pathways. And they can affect specific locations within that pathway that are not always the same from cell type to cell type. So, and part of that depends on where the cell actually came from and its history. So if, well, I'll just stop there before I get too carried away. Oncogenes and tumor suppressors have distinctive functions. And the oncogenes, again, stimulate excessive growth. They frequently have a gain of function mutation, where you have activation of the gene. And importantly, you frequently need only one copy of the gene to be mutated to have an effect. This means it's a dominant effect. On the other hand, tumor suppressors have a loss of function. This inactivates the genes. And both genes must be lost for a cell to become cancerous. This is a recessive trait. On the other hand, oncogenes are dominant. You need to affect only one gene. So looking at the alleles, and the reason I talked about the alleles earlier on was as a prelude to this here.
Let's take a pause here. Aria, are you familiar with dominant versus recessive genes?
Yeah, I've learned about them before in honors bio.
Can you tell us a little bit about, the difference, what's meant by that?
I think dominant is when that trait like always like is taken by the offspring, and recessive is like when it's only taken if it's like in combination with another recessive gene, but it's always like overpowered by the dominant.
Yeah, that's very true. And of course, there's in the real world, there's a little bit of shades of gray, but that's generally true. And a good example is that the brown eyes tend to be dominant, blue eyes tend to be recessive, and you could have one of the parents with brown eyes being a carrier of the recessive gene. And in fact, two brown-eyed people can give birth to a blue-eyed kid if they're both carriers. Now, the role of these genes is actually important. It does matter. Is the mutation on something that's dominant? You know, you could have, for example, potential. You could be at cancer risk if you have a mutation in your recessive gene, but maybe you're okay, right? You never get cancer, but maybe your kids have a chance of having a bad cancer. You know, it can happen, especially if you know you have cancers. There's a little bit of family planning involved, but you know, that's one of the things to factor in is the dominant recessive. Now, that's kind of getting at this loss of function. You know, we're talking about both copies must be lost for the cell to become cancerous. You know, implied in that is that probably the thing that's healthy, the thing that you need, like say the tumor suppressor is you know, it's that brake signal, and it turns things off. So say it's like, say, brake fluid, right? So we're producing brake fluid, which you need to brake your car. Now, as long as one of your two alleles is producing brake fluid, your car has brake fluid and can brake. Maybe not elegantly, maybe it takes longer to stop, but you know, at least you have some basic physiologically relevant amount of that behavior. In the oncogene period, say it is, you know, more of like you go from having things that are normally pieces of wood to being a match that's on fire. Now, all you need is one match that's on fire, and you have like this terrible cascade inside the warehouse, and pretend the warehouse is the cell. You know, you could have just one of them being a dominant driver and having a lot of issues with that. And in the case, that might be, for example, maybe something that's producing gasoline, and it's just producing it in the wrong place. It used to produce it in the gas tank. Now it's producing it on the engine, and like on the top of the engine, and it's just going to catch on fire. You know, it's going to be an issue there. So yeah, I wanted to do that. Paul, I also want to give you some thoughts. We're going to wrap up soon. I'd like to get through signaling pathways and then stop before we get to microenvironment. So just to kind of
I hope we can get that far. We're not going as fast as I had hoped.
Well, yeah, let's see what we get. I think we just have signaling pathways next, so I can yield to you. Go ahead.
All right. You handing it back?
Yeah, go ahead.
Okay. Okay, earlier we had a little bit of a discussion about TP53 and can you force oncogenes to go the other way? We sort of revisit that issue here because the therapeutic goal when you talk about oncogenes is to block or inhibit an overactive protein product of the oncogene. On the other hand, you have a tumor suppressor like TP53. One of the therapeutic approaches is to try to restore the function of the suppressed protein. And again, that's what we talked about earlier. So I want to just reiterate that because it ended up being on being addressed directly on a slide. Okay. So tumor suppressors and oncogenes act as part of signaling pathways. Well, what's a signaling pathway? That is a series of connected biochemical events where you have a signal that is relayed through a chain of activated molecules to trigger a specific therapy cellular response. That was a long sentence. Protein pathways, they control major functions, typically related to cell growth, cell cycle division, variety of different metabolism pathways, and cell death. There's more. And these signaling pathways can affect all compartments of the cell, going from the outside of the cell membrane, to the cytoplasm, through the cytoplasm, to the nucleus, and then to the DNA levels, and then coming out the other side. And dysfunction of these signaling pathways can have an effect directly on diseases and various disease types, and that includes cancer. And another hallmark I might point out of cancers is that they almost always have dysfunctional signaling pathways, one type or another. And I wouldn't be complete if I didn't have a presentation that had way too much information on a slide and nobody can read it, and that's okay. The information is here. Signaling pathways I talked about a little bit before. There's cell cycle, apoptosis. These three here are associated with signal transduction, that is to say, signals of growth factors going from the outside of the cell to the nucleus to control cell growth, and then damage response. All of these signaling pathways have a normal function in cells, either controlling cell growth, controlling death and related events, and a variety of other signaling from the outside of the cell to the inside of the cell. They also have specific effects on oncogenes and the tumor suppressors. And it's typically the opposite. So if you have the effect of oncogenes is actually to promote uncontrolled proliferation. That's what they specialize in. They inhibit cell death. They become constitutively active, which means that they're on all the time in a pathway, even without growth factor signals. Their gas pedal is all the way down, pedal to the metal, going as fast as they can. They don't need control, they think. They maintain constant activation of pathways. Tumor suppressors, on the other hand, are permissive of cell cycle progression. They suppress apoptosis. They act as negative regulators of normal pathways, and so on. But they have specific functions associated with normal cell function and their dysfunction.
Go back to that last slide actually, this slide's good. Maybe you'll cover this. I wanted to give people a flavor, when you get into biology, just generally, and particularly cancer biology, but all biologies, you're going to be, there's like an onslaught of genes, gene names, proteins, protein names, and thankfully enough, there are some things that you become familiar with. So, for me in particular, and maybe for you as well, a lot of this is just recognition, like, hey, I think I've seen that before. And that sense of I think I've seen that before will be overwhelming at the start, but then, the more you see, the more you're like, oh, I've seen this before. mTOR, that's involved in cancer. ERK, that's involved in cancer. Wnt, that's involved in cancer. You kind of start to make these, like kind of small understandings of commonly used pathways. Now, I'll say two things. Well, I'll say a few things. One is that these are the dominant pathways, right? So these are ones that I will say that when you're when a biologist who invented a medication is telling you what does it do, they're probably going to use a mechanism of action somewhere in this pathway. It inhibits part of it, it promotes another part of it, whatever it is. And they're probably going to tell you a narrative that involves these pathways. In a sense, that's fine. These are the driving, dominating pathways. But in another sense, these pathways are running in parallel with each other. Massively in parallel, in fact, and there's other unknown things, right? Like we don't know all the pathways of the cell even to this day, and especially the roles within different tissues. So, do keep in mind that this is a complex orchestration. You could have two pathways in opposition to each other. One is promoting cell growth, one is inhibiting cell growth. There's going to be a winner, right? But without knowing the exact amounts of each and their kind of relative effects, so we don't really know, right? It depends. Usually cell growth signals are turned on and off in well orchestrated ways. Orchestrated meaning maybe there's a buildup of a concentration of something that's signaling for that growth, and then maybe there's an all or nothing turn off that rapidly counter signals. There's supposed to be orchestration there, but these are run massively in parallel. From your perspective, just start to memorize these. You'll see them time and again. You don't have to memorize. What I mean to say rather is just become familiar, just visually be aware. I think I've seen that, and start to build up narratives around them. When you do pick a specialization, say I am a lung cancer researcher, there are certain kind of canonical pathways that are like the main drivers in various diseases. Then yes, then you become a little bit more intimately familiar with that. So again, going from a general biology and a general cancer researcher into something specific, that's when you start to bring on more deeper knowledge. But from your perspective, your goal right now should just be general awareness. Like I think I've seen that before, and I think it was involved in cell survival. And you can look up reference literature, review papers in particular. There's tons of lung review papers. That'll give you a lot of the that'll catch you up in any specific area you're trying to get caught up in. With that, I'll yield back to you, Paul.
Okay, yeah, it's very frequently useful just to think of this thing as one word. This is one word, and this is one word. And the reason for that, again, is because these are just standing for abbreviations. And if you have to remember mechanistic target of rapamycin and use that in a sentence, it doesn't flow well. You have 3 of those in a row, it gets even worse. Every pathway has that same problem. Extracellular receptor kinase. There's this one. I talked about RAS before. So again, for practical purposes, it's easier just to think of these as one word. They describe a pathway. And these are the 3 big ones. These are ones that are the most frequently impacted in cancer. Lung cancer tends to use this pathway preferentially to these other 2. But that does not mean that it does it solely and exclusively. That is not the case. And furthermore, there is overlap and feedback from one to the other. So they go in circles and controlling each other. So it becomes very complicated very quickly. And this is, again, just to give you an overview of what some of the major signaling pathways actually are by their name. The point being that they are crucial for cell growth. They are fundamental components of cell growth regulation. And if you have aberrant activity, you can end up with cancer in any of these. And not only that, oncogenes and tumor suppressors are major components of these things. So it becomes a very highly matrixed and complicated array very quickly. What are the consequences of differences in tumor suppressor genes? We'll talk about TP53. Let's suppose you have a normal cell, and you have one cancer cell with a loss of function. What happens? In a normal cell, you frequently have very low levels of DNA damage. But that low level of DNA damage is corrected by normal DNA repair. If you have a high level of DNA damage, the cell is processed and undergoes apoptosis and dies off. This is a part of the normal functioning of TP53. It controls this damage repair process and apoptosis pathway. On the other hand, in a cancer cell, if TP53 has a loss of function, you have persistent DNA damage. That persistent DNA damage is permissive of a continuing cell cycle and cell division, where you have an accumulation of various gene mutations after each and every individual cycle, leading to chemoresistance, invasion, and metastases. And this accumulation of various gene mutations after the tumor suppressor action is the reason that TP53 is one of the most common and worst in terms of clinical outcome of the mutations that a cancer cell can get. Oh boy. So the tumor suppressors and oncogenes, I think I mentioned, can act through similar pathways. So these arrows here sort of point to a cartoon of a pathway. This pathway starts at the outside of the cell, where you have growth factors interacting with their receptors. Once that occurs, there's a signal that's transmitted from the outside of the cell to the inside of the cell, resulting in a series of phosphorylation events, phosphorus being attached to the protein itself, and that changes its structure and function. It ends up affecting other proteins in a cascade here of different events, all ending up at the level of increased proliferation, invasion, angiogenesis, metastases, and other nasty things. The oncogenes affect various parts of these pathways that can occur at the level of receptors, secondary signaling pathways, or tertiary signaling pathways. Any one of these or all of these can occur in a given tumor cell. In breast cancer, for example, and also lung cancer, these epidermal growth factor receptor gene and the KRAS gene are frequently both mutated. So this signaling pathway is not only turned on, it is really turned on. And then if you can make matters worse, of course, by having a coincidence of a tumor suppressor, which affects exactly the same portion. And now you have a oncogene, or two different oncogenes, and a tumor suppressor affecting the same pathway. You have gone from a situation which was bad to begin with to one which is really, really bad, because the signaling pathways are always and persistently on. And even if you therapeutically, if you try to address one, the cell just shifts over and you start using an alternative salvage pathway. So it runs away from you from a therapeutic perspective.
Paul, we're going to stop there. Can you go back to slide 35 real quick? Before I move on, Olivia, did you have a question?
Oh, I just wanted to say, I don't really want to interrupt, but it may be good if we probably come to a close for today. continue next time, just because we're kind of coming close to like the 2-hour mark. So I don't know if people, especially like rewatching the recording, are going to be able to stick around for much longer.
We're wrapping up at this moment, but before we go, I have some stuff, if that's okay.
Yeah, sure. No problem.
Okay. So this is a very important slide, and I do want to spend some time here. I want to first of all reiterate. Paul, don't leave, please. Go back. I want to reiterate this slide and really to make sure the understanding is clear. The most important thing to understand, for me at least, from starting to get it, is that this is the EGF is endothelial growth factor. So the GF growth factor, right? These are the kinds of things that are triggered in that wound recovery. You know, it's triggered for all kinds of things, you know, cell development and differentiation. But it's the growth factor. Sure, let's take it very simply. During wound, just, you know, you have the epithelial growth factor. Now, where you see that TK, that's the tyrosine kinase receptor. So the tyrosine kinase receptor is a receptor. Now it's binding to the growth factor. Now this is a kind of a simplistic case. What we're talking about are classes of binding agents and classes of receptors. But this is just kind of like a very simple, like, for example case. So you have the epithelial growth factor binding to a receptor. That receptor is a tyrosine kinase receptor. You have the those alpha helices going through the cell membrane, making a channel, and then there's a cause and effect. Now, on that receptor, you see that things are bound. That's GRB, the SOS, the RAS. In fact, there's quite a few others. Those are the protein complexes. And so you have the protein complex. Now, some other conventions here. Below that, you see after RAS, you see RAF, MEK, MAPK. The arrows, by convention, are probably talking about promote. So this promotes that, promotes or activate. I'm going to use in the same way, but it's like a positive activation. The one example on the left, P10, you see that flat line. That's the inhibition line. So some of these, their natural activity is promotion, and some of them are natural activity inhibition. So always keep that in mind. You know, some things are inhibitors, some things are inhibitors, some are promoters. It doesn't really matter which, as long as the orchestration is fine. Now, the point of this is that, yes, the epithelial growth factor does something at the receptor. It changes the shape of things nearby, has that changing of shape. Generally, what it's doing is the shape is changing, exposing a binding region. Something binds to that binding region, and there's probably an enzymatic activity that is activating something else. And that would be like adding the phosphorus group to turn something else on, for example. And that phosphorus group now changes the shape of something, and it's released, and now it can go turn on other stuff. So there's a structural kind of cascade also going on here. I want to talk about this is what the signaling cascade is. You see that there's a left route and a right route, and they're both resulting in the growth stuff at the bottom. So that's that parallelism we were talking about. A lot of what chemotherapies are doing, we're talking about small molecule drugs, kind of like the conventional drug. There's also other things like immunotherapies. We're not going to talk about those, but small molecule drugs, they're going to be probably inhibiting one of these parts. Now, if you were to inhibit P10, you're inhibiting an inhibitor, so it's technically a promotion net effect. But generally, the drug itself, what the drug is actually doing is probably being an inhibitor somewhere along here. So I wanted to go around and do some hypotheticals just to see what, if you guys can trace the logic. So let's say that this is the normal state and the epithelial growth factor is resulting in cell growth. I want to see, Anushka, if we had a MEK inhibitor compound, we gave the cell MEK inhibitors, what would happen? What would your prediction be for cell growth? Would it go up or down?
I'm guessing that it would go down because in the figure it shows like MEK leads to something called MAPK, and that leads to the proliferation. So inhibiting MEK would probably like decrease cell proliferation.
That's right, correct. Thank you.